great for me for the opportunity to speak here. Also, like Kristen, I was trained here at SOC like 16 years ago in Rusty Gage's lab. Okay, so today I'm going to talk to you about our effort in trying to use brain organoids, which is a 3D culture rather than 2D. You just heard from Kristen's work. So usually when you grow iPSCs and try to differentiate and grow those cells under 2D conditions, you have specific cell types. However, if you grow these cells under 3D conditions, you can actually get mini organs to grow. So this is something we have developed in the lab. Actually, it's designed and worked out by high school students to design this spin bioreactors, miniaturized actually, directly adapt to 12 well plates, and you can grow these organoids using these spinning bioreactors and use this platform, and you can see clearly the organoids growing and spinning in the media in these bioreactors. So I'm going to just summarize. Using this platform, we have developed a very robust protocol to generate brain region-specific organoids. In this case, today I'm going to specifically talk about forebrain-specific organoids. And we have shown, since this is already published, I'm just going to summarize that we have observed using this in this organoids. We have shown that in this forebrain-specific organoids, we have human-specific neural stem cell population, which is localized in OSVZ, showing here. And also, we also show that in these organoids, we can actually see neuronal subtypes that can be marked for all 6 cortical layers as markers. And also, they more or less follow this inside-out migration pattern where the superficial layer neurons migrate all the way toward the outer surface. And by looking at the functionality of those neurons in these organoids, we can show that these neurons are functional. They receive both excitatory and inhibitory synaptic inputs. Although it's not shown here, we also show that we have emergence of astrocytes at later developmental stages of these organoids. And if we look at the molecular signatures of these forebrain-specific organoids and directly compare to human frontal cortical tissues, we can show that our late-born up to 100-day organoids is very similar all the way to the end of 2nd trimester of human developing brain. So while this is a very nice, one of the common things that I think for the general organoids field is that when you grow these organoids and they become larger and larger in size, what you usually see is that you start to see a necrotic core in the center because the cell in the center do not get the nutrients and oxygen to support their growth. So usually, as you can see here, because most of the stem cells are localized the inside, and you get this loss of this stem cell population that will limit the further maturation of these organoids. So we developed recently we have developed a method try to overcome this necrosis and improve the survival and health of these organoids. This is what we can get before up to day 70 and day 100, while we do have this stem cell population showing in red here, and also cortical neuronal population showing in green here. This is the size limit about 400 to 500 micron. That's what we can get. If we can limit the cell death or necrosis, we can push further development of the organoids. Now you can see the whole cortical structure can grow almost double in size compared to our previous methodology. And most importantly, we see additional expansion of the human stem cell population as well as the cortical structures. And in this late development cortical organoids, we can see the neural stem cells, the human-specific neural stem cells, localized in OSVZ are still highly proliferative, which is indicated by this proliferation marker Ki67. This is day 120. You can see many active proliferative cells, and 150 you see similar things. In addition, the neural stem cells, or especially the OSVZ cells that is marked by HOPX, they maintain their radial process very nicely, even up to 150 days. And this is actually a publication by Brevenel's laboratory, who is the keynote speaker in the morning, and they show that in human fetal cortical development, there is the maturation process for the neurons is that they gradually, especially the late development neurons, for example, the SETB2, which labels the layer 2 and 3 cortical layers, well, they are diffusely expressed by all neurons at early time points. This is 15 weeks of frontal lobe in human tissue, and you can see a diffuse expression or expression of SETB2 of all cortical neurons. However, over development at 19 weeks, you can see actually restriction or inhibition of SETB2 in deep layer cortical neurons, while the superficial layer neurons maintain the expression of SETB2. This marks the separation and maturation of individual cortical specific layer neurons. So what happens in our organoids if we push them to develop further? So showing here at early time points, day 70, day 100, we also have this mixed population of neurons that express both SETB2, the superficial layer markers, and deep layer markers, TBR1 and the CTIP2. So you can see this population of cells are all mixed together. However, starting from 120 days, you started to see the separation and distinction of these superficial layer neurons and deep layer neurons. This is a more global view of a single organoid. You can see clear separation of the superficial layer and deep layer neurons in our organoids. And also, we know that the superficial, the upper layer cortical neurons, they project, they have these callosal projections that mainly function to connect the two hemispheres, while the deep cortical neurons, usually they are subcortical projection neurons that maintain in the same hemisphere. And you can see here, if we label the axons in our organoids, we can see this horizontal kind of callosal projection of SETB2 positive neurons, while in the deep layer neurons that are CTIP2 positive, they actually have this subcortical projections. So also, this is just give you a more close view of a single neuron that is labeled in our organoids. You can see this is a cortical callosal projection neuron, superficial neuron, that has axons go all the way along the cortical pia surface and has very complex dendritic structures. And if you zoom in, they have nice dendritic spines and synapses and that is co-labeled by synaptic markers. In addition, in this late development cortical organoids, we can also see the maturation of astrocytes. And at the early time points, they have very simple morphology. However, you can see more and more complex morphology of these astrocytes at day 120 and day 150. So in summary, we can push the development of our cortical organoids up to late second trimester, and hopefully this is beyond that time point, we can see specific layer formation in these organoids. So now, what happens can we use this model system to model neurodevelopmental disorders, for example, schizophrenia and other major mental disorders. So previously, we have generated iPSCs from a family with a rare disc1 mutation. I think Kristen gave a very nice introduction that there's this rare monogenic mutations, but with high penetrance. Despite this rare disc1 mutation belongs to that category. In this family, pedigree H, this is an American family. In this family, many members have the mutation, which is a 4 base pair deletion at the end of exon 12. That's a frame shift mutation, caused the early truncation of the proteins in a disc1 gene. And we have generated iPSCs from this family, four members from this pedigree. The parent's father with the mutation and is a major depression patient, and mother, no mutation, is normal. 2 daughters, one with mutation and a schizophrenia patient. The other daughter, no mutation, and it's normal. We also include a control outside the family just to control for the genetic background. So we generate iPSCs from these individuals and then grow organoids from these iPSCs. And if we just look at the general growth of this from these iPSC lines, what we can quantify, for example, the cortical thickness or the progenitor zone thickness of these organoids, the general growth, we didn't see any significant differences from all different patients, different lines, with the mutation or without mutation. There's no difference in the thickness or the general number of progenitor neural stem cells, and there's also no difference if we just look at the cortical thickness or the neuronal cell populations. However, if we look more closely and look at the maturation of these organoids, the neuronal maturation of these organoids, and this is, I've shown you earlier that we see starting from day 120, we started to see separation and distinct laminar layers of cortical structures in these organoids. However, we do not see this maturation in the organoids grown with this disc1 mutation. You can still see the large population actually express the mixed markers for superficial layers and the deep layer markers, SETB2, TBR1, and CTIP2. And again, this is a global view of organoids grown from a control line and from the line with a disc1 mutation. I guess you can appreciate that the cells actually retain high SETB2 expression throughout the cortical plate structures. And a clear view, if we directly compare the expression ratio of SETB2 or to TBR1, you can see over time there's a clear separation again in terms of the expression of cortical markers. There's a decrease of SETB2 expression in deep layer neurons when the increased expression of SETB2 in superficial layer neurons. They mark the different separation of the superficial layers and the deep layers. However, in the mutant disc1 lines, your organoids, you don't see this clear separation. The deep layer neurons still maintain high expression of SETB2. So in summary, what I've shown you here, we can develop, we can grow specific brain organoids using a spinning or approach. And what I don't have time to tell you is that we also have protocols allowing us to develop midbrain organoids, hypothalamus organoids, and hippocampal organoids, and also this allows us to not only to study and compare the basic biology of human brain development and different brain regions, but also this approach will allow us to study and start to understand the molecular mechanisms potentially of neurodevelopmental disorders. In this case, I give you an example, what we can identify layer maturation and the layer specification and early phenotype in these organoids on the schizophrenia or major mental disorder conditions. So with that, I would like to conclude and acknowledge the people. Xu Yubei is a student before I moved to Penn. He's still a JHU student. He's the driving force of this technology in the lab. And Fadi is also an MD PhD student involved in this project. And Chu, actually now, Ki-jung is an assistant professor now in KAIST. And Nam was a graduate student, also important for this project, but now he's a CEO of a company in Maryland. And I also mentioned that the spinning bioreactor actually is designed by 3 high school students when they worked in the lab in the summer. And actually now of course, that's a few years ago. They're now all in college. Max is at Brandeis, and Chris is in Case Western, and William is now in Northwestern. And they are all in biomedical research field, which is very satisfying, of course. And I didn't mention that they designed using the 3D software design, and then actually used the 3D printer to print out what they designed. And of course, we have collaborations, the gene expression, RNA sequencing is a collaboration with Penjin's lab at Emory, and the physiology actually is a collaboration with multiple groups, Brady at the Liber Institute when we were at Hopkins, and Ethan and Lisa are our new collaborators after we moved to Penn. We actually, now we have ongoing collaborations with Ahmed Hoke at JHU to look at the EM structures in our organoids. Thank you.
So we're open to questions.
Hello, I'm here.
Yeah.
So my question is, can you evaluate whether your structural organization of organoids is time-dependent or size-dependent?
So
let's assume you were able to control, to keep your organoids small, but grow them up to 150 days, or you inflate them somehow, and they're going to be bigger, but on day 100. Will they follow the same structural organization?
So in our case, because we specify them specifically to forebrain identity, so the development actually is not size dependent. Although we do have variations in terms of, like, we call this cortical unit, how many cortical units is variable. However, the size of each cortical unit is very consistent, which is not dependent on the time.
Thank you. Another question, have you looked at the functional maturation? Do your neurons and your organoids, they all spike simultaneously, or they spike asynchronously?
Yeah, so we actually showed very briefly, those neurons are actually functional. They have, they do spike, and also I think we have some very preliminary data now to show that in this late development organoids, they actually start to show some sort of the plasticity.
Other questions? Oh, okay.
I was curious, with the family with the DISC1 mutation, does the timing, I don't know if there's data on this, but does the timing of DISC1 expression in organoids correlate with the sort of separation of SAT2B in the layers?
So we have, this one actually there's no good antibodies, but there's a couple of studies to show that actually it is expressed both in neural stem cells and in neurons. So they are quite uniformly expressed throughout the development.
Thank you.
Any others? I have one question. What kind of progress has been made in being able to actually visualize and maybe even trace connections in the organoid between individual neurons and kind of see what their function is? I know it's very challenging. I'm just wondering what kind of progress is being made in that regard.
So, so in general, there are two general category of brain organoids. One is so-called whole brain organoids that is originally developed by a Noblix group, where they have different parts of brain regions. And the one that we have developed here is the brain region specific, so we only have the forebrain. In the whole brain organoids, because they have this brain region, they have different brain regions, but they are randomly distributed. So that makes it hard to look at the connections. Right. In our case, we just started to look how they are connected. Right. However, we cannot see but this is, we showed some potentially look like callosal, for example, and cortical projections. But we haven't looked really in detail, like whether there's, for example, the superficial layer, how they can connect, how they connected to deep layers. That's something we're trying to do right now.
Sure. Okay. If no other questions, the last speaker of the panel, Carol Marchetto.